There's what? There we go. Yeah. I won't look at you. It's fine. No, we'll get through it. It'll be fine. So yeah, welcome to chapter 16. So today we're going to start with gluconeogenesis, and then we'll finish up with glycogen. All right. So here's a list of enzymes that you should be familiar with by now that are participating in either glycolysis or gluconeogenesis or both. So take a couple of minutes and just for each of these enzymes in this list, figure out if it's one, the other, or both. So I'll give you a couple minutes and we'll go over it together. All right, so does anybody want to start us out with which ones are only in glycolysis? Yeah.
Well, I couldn't figure out one of them,
but okay. What do you got? Yeah.
For glycolysis at A.
Mm-hmm.
F.
Mm-hmm.
And H.
Yeah, that's exactly right. So these are the three irreversible reactions in glycolysis. So these enzymes are only in glycolysis. So can anybody tell me for gluconeogenesis? Yeah. Yeah, exactly. So these four enzymes are reversing the three irreversible reactions from glycolysis. They are only in gluconeogenesis. So that leaves us with one last one, which, as you can probably guess, participates in both. So triose phosphate isomerase, or TIM, does a reversible reaction so it participates both in glycolysis and gluconeogenesis. Any questions about that so far? All right, so now we're going back into those reversible and irreversible steps. So these 4 are the list of gluconeogenesis enzymes. Each of them is reversing a step in glycolysis. So, so does anybody want to... So I guess first of all, let's talk about which steps in glycolysis are the irreversible ones. So let's go back to our list. So hexokinase, that's choice H. That's going to be reaction 1 in glycolysis. Phosphofructokinase, that's choice A. That's reaction 3. And pyruvate kinase, that's choice F. That's catalyzing reaction 10 in glycolysis. So these are the 3 reactions that have to get reversed by that list of the other 4 enzymes. So can anybody pick any of these reactions and tell me which enzymes are the ones that reverse them? Yeah. D reverses H. Yes. Trying to pick out my colors. So yeah, so this one reverses step 1. So glucose 6-phosphatase reverses hexokinase. So hexokinase adds a phosphate group to the 6-carbon on glucose. Glucose 6-phosphatase removes that phosphate group from the 6-carbon on glucose. How about another one? Yeah.
G reverses step 9.
Yeah, exactly. So phosphofructokinase, PFK, that's step 3. It's adding a phosphate group to the 1-carbon on fructose. Fructose 1,6-bisphosphatase removes that phosphate group from the 1-carbon. All right, so how about our last one, step 10? We have 2 choices left, right? Yeah.
So FCK and pyruvate carboxylase.
Yeah. Yeah, so these 2 enzymes are both going to reverse step 10. So they, maybe it's easier just to look at those reactions and compare them to each other rather than me describing them to you. So in glycolysis, there is just one step. It's carried out by pyruvate kinase. We're removing the phosphate group from PEP to make it into pyruvate, and we're generating an ATP. To reverse that requires 2 steps. So we start with pyruvate, that's the product. The first step uses pyruvate carboxylase. It uses ATP hydrolysis, and we're adding on this, basically the CO2 onto the pyruvate to make oxaloacetate. Oxaloacetate, keep that in mind, that's going to come up again in the citric acid cycle that we're going over on Monday. So keep in mind that this is one potential entry point to the citric acid cycle. But if we're going on to continue with gluconeogenesis, oxaloacetate finally gets converted back into PEP, which, as you recall, was our starting point for step 10, using the enzyme PepCK. So PepCK is going to use GTP hydrolysis and removing that CO2 that got added on, putting a phosphate group over there to regenerate PEP. So, yeah, does anybody want to take a guess at what class of enzyme pyruvate carboxylase is? So like oxidoreductase, transferase, lyase, ligase, hydrolase, yeah. It is not an oxidoreductase. So oxidoreductases almost always are going to have electron carriers that go along with it, getting oxidized and reduced. Any other thoughts? So, so I can see why you're thinking transferase, because you're you're moving a well, I mean, yes and no. Yeah, you're not transferring it, right? You're doing something else with that other substrate. You're attaching it on, and you're doing so coupled with ATP hydrolysis. So what kind of enzyme couples together ATP hydrolysis with joining together two substrates? Yeah, ligase. Yep. And what about Pepsi K? So for Pepsi K, we're removing this CO2, and we're creating a double bond. What kind of enzyme does that? What's that? Yeah, it's a lyase. So it's a little bit tricky because there's kind of two things going on. But yeah, the part where you're taking off the CO2 and making that double bond, that's the lyase part. There's also the separate part where you're taking the phosphate group off of GTP and putting it onto the oxaloacetate to make PEP. So that you can think of as more of a transferase. But so overall there's kind of multiple reactions going on. So it's a little tricky, but it's kind of a lyase, kind of maybe a transferase. But technically, if you look up like Pepsi K in like a database of enzymes, it's classified as a lyase. All right. So if we look at step 3, for gluconeogenesis, we have fructose 1,6-bisphosphatase. We just mentioned how that's removing this phosphate group. So what kind of enzyme is fructose 1,6-bisphosphatase? Yeah. What's that? What is the name? So yeah, it is a phosphatase because it's removing a phosphate. But of our list of 6 of the classes of enzymes, phosphatase is not one of those. So we have oxidoreductase, transferase, hydrolase is one of them. Yeah, it is a hydrolase, exactly. Yeah, so we're using water. We're adding water, and we're using it to cut this bond. And that is what a hydrolase does. All right, so that brings us to step 1. So we talked about step 1 of glycolysis is hexokinase. It's adding that phosphate group to the 6 carbon on glucose. The reverse of that is using glucose 6-phosphatase to remove that phosphate group. So what kind of enzyme is it? It is also a hydrolase. Yeah, exactly. So just to be clear for the exam, this is fair game. I can give you a reaction and say, what class of enzyme is this? So be ready. All right, let's go back to those questions. Oops. Oh, missed that one. I guess we should start with what is gluconeogenesis? My bad. Hello. Yeah, so we're synthesizing glucose. What's our starting material? Yeah, it's pyruvate. What kind of reaction is it? Is it anabolic or catabolic? Yeah, it's anabolic. Mm-hmm. So, yeah, what makes it anabolic? What are we doing? Yeah. Yeah, we're building up a larger molecule from a smaller one. So, like, how many carbons are we starting with in a pyruvate? 3. How many are we ending with in our glucose? 6, yeah. And also the carbons are they getting oxidized or reduced as we go from pyruvate to glucose? Yeah, it's reduced, yeah. So anytime you have anabolic pathways, we're always going from an oxidized to a reduced. I'll say more oxidized to more reduced, because neither one is completely oxidized or completely reduced. Okay, and what organ is gluconeogenesis mostly happening in? The liver. Yeah, the liver is doing a lot of work for us in terms of metabolism. It's holding on to a lot of our glycogen. It's making a lot of our glucose for us. It's a busy place. Any questions so far about gluconeogenesis in general? All right. So we already did that one because I went out of order. And then we just mentioned which step gets reversed. So pyruvate carboxylase and PepCK reverse step 10 with pyruvate kinase. FBPase1 reverses step 3 with PFK. And G6Pase reverses step 1. If I could spell reverses. Which uses hexokinase. Okay, so let's keep on going. So we talked about glycolysis and gluconeogenesis, and they are in a lot of ways the opposite of one another. Glycolysis is breaking down glucose into pyruvate. Gluconeogenesis is building up pyruvate back into glucose. So if glycolysis is happening, you don't want gluconeogenesis to happen at the same time, because then we're just going in circles and using up a lot of ATP for no reason. So our cells and our bodies have a lot of ways to regulate when one is happening versus the other. And so we talked about this last time for glycolysis in terms of low blood sugar and high blood sugar and that hormonal signaling we get with glucagon and insulin. So there is a template that we'll fill in together that's on the next couple of slides that goes through the hormone signaling and everything that gets activated or inactivated from there. But first, let's go through this whole process together. So when we have low blood sugar, we have signaling from glucagon. Glucagon, through signaling pathways that we discussed a couple chapters ago, we have G protein coupled receptors getting activated. Ultimately, we're making the second messenger, cyclic AMP, inside of our liver cells. That cyclic AMP activates an enzyme called protein kinase A. This is a kinase, so it adds phosphate groups. It phosphorylates its substrates. One of the substrates for pKa is this bifunctional enzyme that contains both PFK2 and FBPase2. So this is regulating that allosteric effector that we talked about a little bit last time called PFK2, called FBP2. So we talked about for glycolysis, PFK1, the enzyme that catalyzes step 3. I cannot talk today. FBP2 is an allosteric activator of PFK1. I know it's a lot of letters, but hopefully this sort of helps to illustrate what's going on. So how do we make FBP2? It's with PFK2. So if PFK2 is active, that means your body's going to make FBP2. In this case, protein kinase A, when it phosphorylates this bifunctional enzyme, it's going to do not that. It's going to activate FBPase2, and it's going to inactivate PFK2. So this means you're going to make less FBP2. If you make less FBP2, that means PFK1 is not working. The opposite, the enzyme that opposes it, is working. We just talked about what opposes step 3, FBPase1. So FBPase1 is activated, which means gluconeogenesis is going to happen. So this starts with glucagon signaling because you had low blood sugar. A bunch of regulatory enzymes get activated, in particular this very important bifunctional enzyme that determines whether or not we make FBP2 or not, and that's going to affect whether or not we go into gluconeogenesis or glycolysis. So for glucagon, we go through this whole long pathway, and gluconeogenesis is going to happen in the liver. For insulin, that's the opposite, right? So if you have high blood sugar, that's when you get insulin release. Insulin is going to activate a different signaling pathway using those receptor tyrosine kinases that we mentioned a couple chapters ago. Eventually, that receptor tyrosine kinase is going to activate this second messenger called PIP3. PIP3 is going to eventually activate a different protein kinase B, or PKB. So glucagon turns on PKA, insulin turns on PKB. PKB has the same function as PKA. It's also a kinase, so it also adds phosphate groups. But PKB has different targets. So PKB is going to phosphorylate a phosphoprotein phosphatase. And this one that we are interested in is called phosphoprotein phosphatase 1, or PP1. So PP1 has the opposite function of PKA. So we talked about how PKA is adding phosphate groups to this bifunctional enzyme. PP1, it's a phosphatase. It removes phosphate groups. PP1 is going to remove the phosphate group from this bifunctional enzyme. So this is going to have the opposite effect of PKA. So in this case now, the PFK2 portion is active. So this enzyme is going to make more FBP2. And when there's more FBP2, which enzyme is getting activated? PFK1, yeah. And if PFK1 is activated, which metabolic pathway is going to happen? Glycolysis, yeah. So yeah, this is I think, pretty good summary of what you need to know about how all this stuff is getting regulated. So you should be familiar with all of these steps. What is phosphorylating what happens when it gets phosphorylated. So now let's go and, yeah.
So is the name of the bifunctional enzyme FBP2? Those two combine equal FBP2?
So it's usually, I'll just call it the bifunctional enzyme containing FBPase2 and PFK2. It doesn't really have a name aside from that. It's kind of goofy. But they're connected to each other so that when one is activated, the other is inactivated. And they're physically together on the same enzyme. And so unfortunately, it's just called the bifunctional enzyme containing FBPase2 and PFK2. Yeah, we can come up with a better name for it for extra credit. Okay, so now that we know this, let's go and try to fill in the blanks in that paragraph. So let's start with low blood sugar. If there's low blood glucose, which hormone is going to get released from the pancreas? Yeah, glucagon. Glucagon is going to bind to its receptor, and it's going to make eventually which secondary messenger? Yeah, cyclic AMP. When cyclic AMP is produced, which enzyme is getting activated? Yeah.
PKA.
Yeah, PKA. There's some other stuff in between, but we care right now about PKA. And so what enzyme gets phosphorylated by PKA? Yeah, so it's the bifunctional enzyme containing PFK2 and FBPase2. Sorry about the name. Or, yeah, I guess we can just write the bifunctional enzyme there and fill in the other ones on the other side. Forgot what that's for. That's a little easier. Okay, so which one gets activated and which one gets inactivated when it's phosphorylated?
FBPase2.
Yeah, so FBPase2 is activated, which means that PFK2 is inactivated. So does that mean we get more or less FBP2? Yeah, less FBP2. And we know that FBP2 is an allosteric effector. Which enzyme does FBP2 activate?
BPase.
Almost. The opposite one.
What's one PFK?
Yeah, it's PFK1. And if PFK1 is getting activated, what is getting inhibited by FBP2? Yeah. So it's not the bifunctional enzyme is what's making FBP2. But now I'm asking for FBP2, it's an allosteric effector of both PFK1 and another enzyme that has the opposite function of PFK1. Yeah. Yeah. Because the whole idea with glycolysis versus gluconeogenesis is if you turn on one, you have to turn the other one off. So FBP2 is turning on PFK1, which is going in one direction, so it's going to turn off FBPase2, or FBPase1, which is going the other direction. So now that we know FBPase2 is there's less of it being made in low blood sugar, and FBPase2 is something that activates PFK1, so which metabolic pathway are we upregulating right now? Yeah. Gluconeogenesis, yeah. I know it's a lot, and it's a lot of letters and numbers that all sound very similar. If it helps, we can use their full names. They're just very long. So we can call them phosphofructokinase 1 and 2, and fructose 1,6-bisphosphate, and fructose 2,6-bisphosphate, and fructose 1,6-bisphosphatase, and fructose 2,6-bisphosphatase, but our hands would all get very tired from writing all those names. But that being said, are there any questions about how this works? Okay, so let's try it for high blood sugar. So I'll give you a couple of minutes to fill this one in on your own, and then we'll go through it together. Alright, so let's start with high blood sugar. So high blood glucose really results in the release of which hormone from the pancreas? Yeah, insulin. So after insulin binds to its receptor and it starts a signal transduction pathway, which enzyme is going to get activated? Yeah. PKB. Yep. So PKB is a kinase, so it phosphorylates and activates which enzyme? Yeah. So not yet. So that enzyme gets phosphorylated by PKA. PKB has a different target. What is the target of PKB? Yeah, PP1. Yeah, sorry. So PP1 is a phosphatase. It dephosphorylates. And what does it dephosphorylate? Yes, the bifunctional enzyme. So our bifunctional enzyme has PFK2 and FBPase2. Which one, when it's dephosphorylated, which one is activated and which one is inactivated? Yeah. Yeah, PFK2 is activated, FBPase2 is inactivated, which is the opposite of what we saw with our low blood sugar. So if PFK2 is activated, do we get more or less FBP2? More, yep. And FBP2, as we know, is the allosteric effector for one of the enzymes in our metabolic pathway. Which one is activated by FBP2? Yeah, PFK1. And so if it activates PFK1, what's getting inhibited? What does the opposite of PFK1? Yeah, FBPase1. So if PFK1 is activated, which metabolic pathway are we going to activate, upregulate? Yeah, glycolysis. So I know that, again, this is a lot of letters and numbers. If it is helpful for helping you remember what's happening when PFK2 is activated, so is PFK1. For the opposite, when FBPase2 is activated, so is FBPase1. So the way that this is worded is just as the opposite, which is why it looks like it says inhibits. But let's actually go back to the picture. It's probably easier. So if PFK2 is active, PFK1 is active. If FBP2 is active, FBPase1 is active. So PFKs go together, FBPases go together. Then you just have to remember which one gets activated when there's a phosphate added and which one gets activated when you remove the phosphate. Yeah, so for these pathways, if you're having trouble kind of remembering what's happening whenever one of the PFKs is activated, so is the other one. So if PFK2 is activated, it makes more FBP2, and that turns on the other PFK, PFK1. Whenever FBPase2 is activated, it turns on FBPase1 because there's less FBP2. So that's maybe something helpful.
So PFK1 is when phosphates removed, kind of.
So,
and the base is when it's added.
Yeah, so the FBPase2, yeah, is activated when the phosphate is added. Yep. When the phosphate is removed, that's when PFK2 is activated. So in a way, that's kind of the opposite. You add a phosphate group to the enzyme, and the enzyme starts taking away phosphate groups from its substrate, which is a little bit confusing, but that's just how it works, unfortunately. What's that?
It's still active just to the both enzymes are still active just to allow some stuff until the phosphorylated. Because once it goes there,
Right. I mean, I think in general with anything in biology, nothing is always 100% active or 100% inactive. There's always an equilibrium. So you could say it as phosphorylating this bifunctional enzyme shifts the equilibrium towards FBPase2 being activated, more active than the other. Dephosphorylating the bifunctional enzyme shifts the equilibrium the other way so that the PFK2 is more activated compared to the FBPase2. But nothing is 100% in biology. In terms of on the exams and quizzes, you'll probably see it both ways, like inactivated or activated, or more activated, less activated. It's kind of the same idea, just one's a shortcut, I guess, or a little bit less accurate. Any more questions about gluconeogenesis or this regulation, hormonal regulation? Okay, so let's find our questions again. Okay, so if we're good on gluconeogenesis, we're going to move on to glycogen. So glycogen metabolism, I'll click on here, make sure I didn't go too far. Okay, so So take a moment and think about these enzymes. All of them participate in either glycogen synthesis, glycogen breakdown, or both. So take a moment and figure out which ones are for synthesis, which ones are for breakdown, which ones are for both, and then we will figure out the order. Alright, so can anybody tell me which ones are used only for glycogen synthesis? You don't have to name all of them, just any of them. Yeah. Branching enzyme, inorganic pyrophosphatase. Yeah, UDPG pyrophosphorylase. And glycogen synthase. Yeah. Does anybody have the ones used only for breakdown? Yeah. Glycogen phosphorylase. Debranching enzyme. Excellent. So that leaves us with one left. So phosphoglucomutase is used in both. Alright, so let's start to put them together. So we have, what's our starting material for when we're going to synthesize glycogen? Is it just regular old glucose or is it a modified glucose? Yeah. Yeah, glucose 6-phosphate. I'll write as G6B. And what do we need to make it into before we can start going through glycogen synthesis? Yeah, that's the enzyme we use. What is that enzyme doing with glucose 6-phosphate? Yeah, glucose 1-phosphate. So we're PGM, phosphoglucomutase, moves the phosphate group from the 6-carbon to the 1-carbon on glucose. Now the glucose is ready to start becoming glycogen. So what happens from there with glucose 1-phosphate? What enzyme are we using and what's happening? Yeah. Yeah, so we make I can't spell either, UDP glucose, which you can also call UDPG. And so for that we need another substrate involved, right? We need UTP. We're taking so we're taking part of that UTP, we're sticking it on to the glucose 1-phosphate, and at the same time we're hydrolyzing that UTP into, and we're hydrolyzing off the inorganic pyrophosphate. So which enzyme does that part? Sticks the UTP on to the glucose 1-phosphate, releasing the pyrophosphate. Of all the enzymes in this list, which one sounds like it does that? Yeah. Yeah, exactly. So this is UDP glucose. Maybe I'll change colors. Oops, not pyrophosphatase. So UDP glucose pyrophosphorylase is sticking the UTP onto the glucose and releasing the pyrophosphate. And what is happening to pyrophosphate? Yeah. Yeah. So this cleavage also helps to lend energy to this whole reaction that's activating the glucose molecule. So with these two enzymes, now we have this sort of activated glucose, this UDP glucose. What happens to the UDP glucose? Yeah. UDP. No, what happened to the UDP glucose? The UDPG, yeah. Yes, yeah. So, yeah, it's kind of like weird how to write it, but so you can sort of, we're adding on to glycogen, and we lose the UDP, and we use, you told me which enzyme, right? Glycogen synthase. So glycogen synthase is adding on these UDP glucoses, or just the glucose, onto an existing glycogen molecule. what type of glycosidic bonds is it making? Yeah. Yeah, alpha 1,4. And then we also, I think you also mentioned the branching enzyme. So we have our branching enzyme can come along What kind of glycosidic bonds does the branching enzyme make? Yeah, alpha 1,6. Yeah. All right, so that's synthesis. So let's try to break it down. So what is the first step when you're breaking down glycogen? Yeah, we have our debranching enzyme. There's not really a great way to write that. And then after our debranching enzyme, oh, and which kind of bonds is our debranching enzyme removing for the most part? Yeah, the alpha 1,6. There's a little bit more to it than that, which is in the slides, but it's kind of the idea behind debranching. And then how do we remove the glucoses from the glycogen? Yeah. Yeah. So we have our debranching enzyme. I'm just trying to decide if I wrote this right. Maybe I'll write this as N+1, and then I'll just write this as regular glycogen. Okay, so we have, so we add in our inorganic phosphate, and we have a glycogen that gets turned back into, that doesn't really seem right. There we go. So, yeah, and that uses glycogen phosphorylase. Not the best diagram. There's a nicer one in the lecture slides, but you get the idea of the main enzymes involved and sort of what's getting added and removed. And then we also know that the glucose 6-phosphate that gets made with PGM can enter into glycolysis, right? Because glucose 6-phosphate is our second metabolite in glycolysis. Any questions about that so far? All right. So, so now let's start looking back into regulation. So we know that all of these processes that we've talked about so far with glucose metabolism, glycolysis, gluconeogenesis, glycogenolysis, and glycogen synthesis are all either breakdown or synthesis pathways. They're anabolic or catabolic. So first of all, let's just go through and categorize each one of them as being for breakdown or for synthesis. So is glycolysis a synthesis or a breakdown pathway? Yeah, breakdown. And what about gluconeogenesis? Yeah, synthesis. So these are the opposite pathways for glucose. One is breaking down glucose, one is building up glucose. How about glycogen? Is glycogenolysis breaking down glycogen or building up glycogen? Yeah, it's breaking down. And what about glycogen synthesis? It's in the name. Yeah, it's a synthesis pathway. Yeah. So on the right, we have boxes. So all of these 4 different pathways are regulated by concentration of glucose and concentration of ATP. So availability of fuel or reactants and availability of energy currency. So take a moment and fill in the boxes. So are all these 4 processes going to happen when glucose and ATP are at high levels or low levels? So take a few minutes and then we'll discuss it together. All right, so let's start with glycolysis. So what are glucose levels high or low when glycolysis is going to happen? Yeah, high. Yeah, you need a lot of starting material to get the pathway going. And what about ATP levels? Yeah, low. Yeah, so glycolysis is going to happen when you need to make more ATP. High levels of ATP will inhibit glycolysis. How about gluconeogenesis? Yeah. Yeah, exactly. You only want to go through the trouble of making new glucose when you don't have a lot, because it's relatively energy expensive to make more this way. And what about ATP levels? Yeah, high. Yeah, so you need to use a lot of ATP to go through gluconeogenesis, so you're only going to do it when there's enough ATP around. You can also think of it as the opposite. So they will occur during opposite environmental conditions, which makes sense. You don't want to be breaking it down while you're building it back up, because that's pretty wasteful. All right, so how about glycogen breakdown, glycogenolysis? Yeah. Yeah, exactly. You're breaking down glycogen to release glucose, so you're only going to do it when there's not a lot of glucose. And how about ATP levels? Yeah, also low. Yeah, you're breaking down, you're releasing the glucose so you can use it for glycolysis because you need to make more ATP. So how about glycogen synthesis? Yeah. Yeah, high levels of glucose. If you have a lot of extra glucose, you may as well store it as glycogen. And what about ATP levels for glycogen synthesis? Also high. Yeah, glycogen synthesis requires energy input in the form of that UTP. So you're only going to do it if you have relatively high energy levels in your cell. And also it is the opposite. So the breakdown and the synthesis are going to occur under opposing environmental conditions so that you don't have them both happening at the same time. Any questions about that? Yeah.
So I'm a little confused because like glycogen synthesis usually happens when, when you said there's high glucose. So like why would you, if there's already high glucose, why would you be actually not.
Okay.
If there's already high ATP.
Yeah.
Wait,
So you kind of have two options. If you have high glucose, you can use it to make glycogen, or you can use it, you can break it down to in glycolysis to use it to make ATP. And so glucose is high, the decision you make is how much ATP is around. If there's not a lot of ATP, you use all that extra glucose in glycolysis. If there's already plenty of ATP, you have enough energy, so you make the decision to store it as glycogen. Any other questions? All right. So let's think about each of our 4 pathways that we know so far. And take a minute and think about what is the main regulatory enzyme in each of these pathways. And then we'll talk about it together in a minute or two. All right, so let's start with, I guess, any of them. Anybody want to throw me a regulated enzyme in any of these 4 pathways? Yeah.
Glycogen breakdown would be glycogen phosphorylase.
Yes. Glycogen. Phosphorylase, which you can call GP. Any other ones? Yeah.
Oh, glycogen synthesis would be glycogen synthase.
Yeah, glycogen synthase. And these 2 enzymes are essentially doing the opposite functions of each other in glycogen synthesis versus breakdown. They're removing or adding those alpha-1,4 glycosidic bonds. So they both are getting regulated so that you're only making the bonds or only breaking the bonds at any one time. All right, how about for the other 2? Yeah. Yeah, yep. PFK1, phosphofructokinase 1, PFK1. I think I spelled that right. Yeah. Yeah, it's the 3rd step in glycolysis. How about gluconeogenesis? Yeah.
FBPase1.
Yeah, FBPase1. I don't really want to write that one out, so we'll just call it FBPase1. And again, these 2 steps are opposing each other. The 3rd step in glycolysis is only goes, what's the word for it? It's irreversible. It's done by PFK1. The reverse direction is done by this other enzyme, FBPase1. And they are doing opposite functions. PFK1 is adding a phosphate group to the 1 carbon on fructose. FBPase1 is removing that phosphate group from the 1 carbon. Any questions about that? All right, so now we're going to go through our same little thing that we just did with glycolysis and gluconeogenesis. So we're going to talk about regulation of glycogen metabolism in response to low blood sugar and high blood sugar. So let's go through those fun little animated slides first, just to refresh ourselves. If I can find them. I went past them clearly. There we go. All right, so we'll start with glycogen breakdown. They're big, so they're on 2 slides. So we already know the punchline. Glycogen is going to get broken down at the end of this. This one starts with glucagon. So glucagon is our sign of high or low blood sugar. Yeah. Low blood sugar. Yeah. So we already know that when there's low blood sugar, we need to release the glucose. So it makes sense that glucagon is going to end up breaking down glycogen for us. So we already know the beginning of this. So glucagon activates a signaling pathway that results in the activation of PKA, protein kinase A. We know that protein kinase A, it's a kinase. It phosphorylates its targets, its substrates. One of the targets of PKA that is relevant for glycogen breakdown is phosphorylase kinase. So phosphorylase kinase is regulated in response to PKA signaling. PKA adds a phosphate group, and that's going to activate phosphorylase kinase. When phosphorylase kinase is active, itself, it is also a kinase. It also adds phosphate groups to its targets. One of the targets of the target of phosphorylase kinase is glycogen phosphorylase, which we should be familiar with from, I believe, chapter 12. And we know that phosphorylase kinase adds phosphate groups to glycogen phosphorylase, and this also activates it. When glycogen phosphorylase is activated, we just talked about how that is going to break down glycogen by removing those, or by cleaving those alpha-1,4 glycosidic bonds and releasing glucose 1-phosphate. On the other hand, we can also start with insulin signaling if we have high blood sugar levels. We also already talked about how insulin starts that signaling pathway that eventually results in activating PP1, our phosphatase. Phosphatases have the opposite function of kinases. They hydrolyze phosphate groups. So PP1 is going to remove the phosphate group from phosphorylase kinase. It's going to inactivate it. And because phosphorylase kinase is not activated, it's not going to be adding phosphate groups to glycogen phosphorylase. At the same time, PP1 also removes any phosphate groups from glycogen phosphorylase. And that makes it less active. When glycogen phosphorylase is less active, that means we get no glycogen breakdown. So that's how those two hormones work in response to, or in regards to glycogen breakdown. And then now let's take a look at how they work for synthesis, which is pretty much the opposite. So once again, we can start with glucagon. That's our sign of low blood sugar. We already know that glucagon activates pKa through our signaling pathway. We already know pKa activates phosphorylase kinase. When phosphorylase kinase is active, it also phosphorylates another target. The other target is glycogen synthase. So we are adding a phosphate group to glycogen synthase, but when glycogen synthase is phosphorylated, it becomes less active, which is the opposite of how glycogen phosphorylase responds to being phosphorylated. When glycogen synthase is less active, we are not going to synthesize glycogen. And then for insulin signaling in response to high blood sugar, we basically have the opposite. So insulin, as we know, activates PP1. PP1 is going to remove any phosphate groups from phosphorylase kinase, making it inactive or less active. PP1 also removes phosphate groups from glycogen synthase. When glycogen synthase is dephosphorylated, it is now active. So it is going to start synthesizing glycogen. So those are the big enzymes that we're regulating. So not just glycogen phosphorylase, also glycogen synthase and phosphorylase kinase. Those are the big ones. So now that we discussed that a little bit, let's go and answer these questions. So now we're going to talk about glycogen regulation in response to low blood sugar. So maybe take like a couple of minutes and fill this in on your own, and then we'll go over it together. Okay, so let's start at the beginning. So low blood glucose results in the release of which hormone? Yeah, glucagon. And which kinase gets activated by glucagon? Yeah, PKA, yeah. So, PKA phosphorylates which enzyme? Yeah. Yeah, phosphorylase kinase. Which you can also call PK. Just be careful because PK can also mean about 5 other enzymes that we discussed during this class. So PK phosphorylates, so when phosphorylase kinase gets phosphorylated, is it activated or inhibited? Yeah, it's activated. And then which enzyme does phosphorylase kinase phosphorylate? Yeah, glycogen phosphorylase. Or GP. Is GP activated or inhibited when it gets phosphorylated? Yeah, activated. Okay, so if glycogen phosphorylase is activated, what is happening with glycogen breakdown? Yeah, it's upregulated. Okay. So, and hopefully this makes sense because remember this all started with low blood glucose. So we need to break down glycogen to release some glucose. All right, so let's think about the other direction. So PKA and PK also phosphorylate another enzyme. Which enzyme is that? Yeah, glycogen synthase. Or GS. And is GS activated or inhibited when it's phosphorylated? Yeah, it's inhibited. So what does that mean for glycogen synthesis? Yeah, it's downregulated, which also makes sense. We started with low blood glucose. There's not a lot of glucose around, so we can't really use it to make more glycogen. Okay, so let's do the other way, high blood sugar. So take a couple minutes and fill this in, and then we'll go over it together. Alrighty, so let's get going with high blood glucose. So, which hormone gets released when there is high blood glucose? Yeah, insulin. And which kinase gets activated by insulin? Yeah, PKB. So PKB phosphorylates which enzyme? Yeah, PP1. And when PP1 is phosphorylated, is it activated or inhibited? It's activated. Alright, and PP1 is a phosphatase, so it dephosphorylates which enzymes? Yeah. Yeah. Phosphorylase kinase and glycogen phosphorylase. And when those enzymes are dephosphorylated, are they activated or inhibited? Yeah, they're inhibited. Okay. So, what does that mean for glycogen breakdown? Is it upregulated or downregulated? Yeah, it's downregulated. Okay, so PP1 also dephosphorylates and activates and does something to another enzyme. Yeah, glycogen synthase. And is this enzyme activated or inhibited when it's dephosphorylated? Yeah, it's activated. So what does that mean for glycogen synthesis? Yeah, it's upregulated. And this should also make sense. So we started this whole thing out with high blood glucose. High blood glucose means you have extra glucose. So it's a good time to not break down glycogen. It's a good time to make glycogen with all the extra glucose that you have. So, any questions about glycogen synthesis and breakdown or gluconeogenesis? Okay, so in that case, that's all the questions I have for today. So I just want to remind you that tonight, quiz 11 is due, if you haven't done it yet. 1 week from today, next Thursday, the 7th, I think, is when quiz 12 will be due. Quiz 12 is on 15 and 16, which we just finished today. And the following Thursday, the 14th, is exam 3. Your final draft of your project is due next Friday, which I believe is the 8th. And I am almost done getting feedback. Sorry, it's a little bit delayed, but by Friday, like by tomorrow, I will get feedback to all of you on your project updates and rough drafts that you submitted. Yeah, if you have any questions for me, I have email, I have office hours right after this. Yeah, you have a question?
Extra credit for the last quiz that was on Monday. I don't think it came through. It just stopped at 100. That's supposed to get that?
No, I think I think I need to go through and manually do those. I'll do that. Yeah, sorry. Thanks for reminding me. Any other questions? All right, then you are free to go.